using neutron bombardment in a nuclear reactor. And that turns some of the potassium into a different type of non-natural argon called argon-39, which we can then measure. So we can measure the parent and the daughter at the same time. And yeah, I run that in my lab at the University of Nevada, Las Vegas.
I think you're tugging.
It's a very
Roger.
detailed method, but it produces nice ages of these kind of specific rocks. And it's one of the only ways to date these type of rocks.
That's cool, thanks.
Do you think it's possible we can move more westward to see on the other side of this cliff face if it's more life or less?
Sure.
SPL, this is Data Lab. Just a heads up, I had to restart the process that sends data to Grafana a few minutes ago, so should be fine now, but I don't know, be more suspicious of it than normal.
Roger, thank you, Tim.
Let me still bubble real quick.
Roger.
Oh, broken off needle.
So 95 million years ago was kind of like prime T-Rex period, right?
Actually, it's prior to T-Rex. T-Rex didn't come about until about 66 million years ago. The first T-Rexes appear basically right before the extinction event.
Really?
But it is mid-Cretaceous. Okay. It's prime Spinosaurus territory, which is the coolest dinosaur, as we all know.
Here's a question for our scientists. Will any of the water samples be analyzed for microplastics?
We aren't actually planning to analyze them for microplastics on this cruise. It takes a little more specialized equipment than we have to process for microplastics on board. But it is something that we periodically get researchers looking for out of the water samples. One of the issues we have is like the Niskin bottles themselves are made out of plastic, and when you're looking for microplastics, you really want to isolate from as many other potential cross-contamination sources of plastic. You could still process them for it, but yeah, right now we're just not set up in the lab to do that.
Those almost look like little tracks on the side of the rock there.
Yeah, I think those are from some of the worms, I think. Eat their way along the rock face.
Cool.
Also, ferromanganese doesn't grow on top of sediments. So if you have sediments in a crack, the ferromanganese will just grow around it and it'll get more pronounced, the crack, and just kind of remain through time.
You want to come on up on the delta a bit there, please? This entire area looks like dark volcanic rock. Where do the lighter colored sediments all come from?
That's the marine snow. So it's carbonate little shells and poop and other things from the critters on the surface that just kind of settle through time. Seems like the deposition rate in this area is pretty low. Some areas in the Kingman region we were a few years ago, and it was just, you can see it, it looked like almost like a blizzard in some areas when there's so much marine snow and so much sediment.
Which way are we moving again right now?
We're starting to go 015. With a little bit of swing left over from the 060.
Gotcha.
Okay, yeah, out in front here, and then I'll switch you over with you if you want to fly for a while.
Oh, no, that's so fine. I'm good. You don't want to fly
That's
tired,
a nice
you know
steep
what
feature.
Sorry?
I mean? So you don't want to fly tired. I don't want to fly tired. It's dangerous. Yeah.
This has been a great example of just how much variability you can get. We got into the little patch for a little while where suddenly we had more corals and sponges and then popped right back out of it. Probably just something about the local hydrology right in that zone, funneling water to them in a way that they find the most beneficial.
Come down. Come down. There you are. Also,
Sorry, Becca. We might have low visual contact for a minute, just while we get ourselves situated for this upcoming move. Be about 2 minutes.
are you coming up or coming down? Okay. Have you ever been in a late watch where everyone just like somehow was able to communicate via yawn? You know what I mean?
I feel like that would be highly contagious and the message would get lost and
Yeah.
within all the yawns.
I think it would eventually become detrimental too.
You yawn and then they're like, oh wait, I'm sorry, was that 050? No, 250? You know, then.
Well, no, but like you've been going for so long, you understand each other. You know what I mean?
Another language.
Like one of those things you can only understand it while you're doing it too.
Another little chrysogorgia on the left of the screen.
Oh yeah.
That's an interesting thought.
Go ahead and do a quick zoom here, please.
Zooming in.
That should be good. Wee little guy. All right, full wide, please.
Full wide.
I feel like the first thing that Dan's going to do when he gets in here is throw that plate. With
Get
all
this
the,
out of here.
all the line in the way.
Okay, so do we want to keep moving across this ridge to the other side still, or are we centered up enough, or?
Yeah, I think we keep moving to the other side of the ridge.
Okay, so just keep moving in the same direction?
Yeah, yeah.
Okay.
As we go towards waypoint 3, we just kind of head in a slightly northward direction.
Yep.
Yeah, perfect.
Bridge nav. Oh. 100 meters, 025.
Good. Come on up a bit there, please.
Yeah, I don't know why it's going slow. That's odd. Huh?
There's a nice chrysogorgia right above the lasers.
Seems to be like the only type of coral we're seeing, right? It's the chrysogorgia and then earlier on the bamboo coral, but
Yeah, we had a few bamboos and then it's been all chrysogorgia all the time for the last hour and a half or so.
A little dandelion looking.
oh, sponge. Tubing.
Gnarly looking landscape.
The more fragment of dead sponge laying there on the bottom, I think that's more of that Pharia.
Yeah, there's more up ahead too.
What a plot twist if you found out that all of this is just dead dead sponges.
Manganese encrusted sponges.
A Pompeii-like event.
Go ahead and fish on in there a bit, please, there, video.
It's moving in.
Oh. What's this?
A little tighter.
And more for All right, full wide, please.
Full wide.
And another chrysogorgia.
It's the same one.
I'm going to go out on a limb and say I'm pretty sure those bottle-brushed chrysogorgia are Chrysogorgia geniculata.
The next one you see, you want to close up on the polyps so you can see what that helps you with the ID.
I don't think that would help any more for me. Chrysogorgia is not a group that I'm that much of a specialist in. But that's looking at all the guides, that's the most common with the bottle brush form with the polyps that go all the way down to the base rather than a clear stalk.
I see. Roger that.
There's a little sediment valley filled with nodules right below us.
Yeah. It's kind of neat.
Yeah.
A little bathtub full. Well, we've come up about 300 meters in the elevation so far. That's pretty good.
It would be a fairly intense hike if it were above water, I think.
What's that?
I was thinking it would be a pretty tough hike if it were above water.
A small coral to the right.
Little bamboo coral, yeah. Gonna go ahead and push on in there a bit, please. Yeah. Not a lot of sea stars or anything down here either. You got any more zoom in there on the polyps?
I've seen just a few of the brachyngeids, but not a lot of anything else.
Awesome. Full wide, please.
Full wide.
How How do they stay stuck to the rock like that?
So they've got a holdfast that is basically that kind of wider disc at the bottom that they generate their own kind of bio-cement and attach to the rock with.
That's really cool.
Or you can, sometimes, well, they can be very, very tightly cemented. We've actually picked up whole rocks by picking up just a broken off stalk, and the rock comes with the stalk.
Oh my goodness. So strong. There's a whole really cool field of bio-inspired adhesives that are based on mussels. How mussels can hold really well to rocks or anything that withstand a lot of waves. That and sandcastle worms, both of which have inspired some interesting adhesives for use in underwater environments. I wouldn't be surprised if corals would also do the same for bio-inspired adhesives.
Look like another long bamboo coral to the right, and then we got some sponges straight ahead.
And I guess bottle brush
no.
corals.
No, those are corals.
Okay, yep. More chrysogorgia.
Nice little colony. I'm gonna go ahead and do a partial zoom here.
Partial zoom.
Very cool. Okay, full wide, please.
Full wide.
This one's really nice. Go ahead and get a tight shot here, please.
Full zoom.
Oh, wow.
Sorry, I'm gonna push it around for a second.
It's a really nice shot there.
There you go. Oh, she came up with a breeze too.
Actually, I'm gonna come partial wide, please.
Partial wide.
Set you about that.
How's that?
That was great. I'll set us up a little bit better. All right, go ahead and push on in there again, please. Okay, full wide, please.
Okay, full wide.
Okay.
Nice shot of those delicate bell-like polyps spaced out along each branch.
Rage nav, 100 meters, 040.
The branched bamboo coral just going off to the right.
Oh, yeah. Looks like there might be actually 2 stalks, hey? Do a quick zoom there, please.
Zooming in.
That's good there.
Holding here.
Oh, you're right, yeah.
Oh, yeah.
Full wide, please.
Nice. Thank
Full
you.
wide.
Thank you.
Yep.
Sneaky little coral, chrysogorgia, hiding in the rocks to the left.
They can be so hard to spot because they just can be these floating, delicate puff balls that just don't stand out that much against the background.
Yeah. We are starting to see more corals, though, as we're both going upslope, and we're on the northern side of the slope now. I don't know if that's helping or not, but we're definitely seeing a bit more.
So Bob Ballard wanted to say thank you for the great frame grabs of the dead sponges. He's saying that we've seen some living attached ones, dead attached ones, and then those that fall off the rocks, leaving a stain where they were attached to the rock. You get a relative age by their sediment coverage over time, but the real old ones are coated black by manganese oxide. Since you're diving on a mid-Cretaceous seamount dating back 115 million years, he's interested in seeing if their taxonomy has changed over time. So just wanted to give you that explanation there. Let's see if we have any more questions coming in.
Very
so
cool.
Here's a question about the corals. Do the colonies all gather in larval stages and then grow, or do individuals continue to join in the colony?
So the whole colony will be based off a single settling larvae. And then one thing that can happen actually in some species of coral is you can have what's called polyp bailout, where individual polyps can leave the coral and go start a different colony. You can end up with then clones within an area. It's relatively poorly documented, but there have been some well-documented examples. But yeah, each of these individual corals will have started from a single larvae and then grown into the full colony. Sometimes in an area where you see all one species and they're all about the same size, what we think is happening is that you've had kind of a mass settling event where a bunch of larvae from the same spawning event have all landed in the same area and started to grow. But with deep sea corals in particular, very little is known about their reproductive biology and how and where their larvae move.
Can we have a zoom on that sea pickle?
Yeah, let's do it.
Let's catch it on.
Sea pickle. All right, go ahead and push on in there, please.
Zoom it in.
I'm gonna make us seasick for 1 second by bumping around the camera. Bump up.
A bit farther.
The camera's, as you can tell, the pan and tilt is a little jumpy. It looks like a strawberry.
Yeah, does anyone know if this is one Liz would want to sample?
As I recall, she wanted any that were big enough, but was somewhat species agnostic.
All right.
Any that she could digest the gut out of.
So do you want to try for this one?
We're
Or
kind
is it
of...
too small, too hard?
Oh, no, it's just that we're about 100 meters laid back from the ship, maybe less, maybe 70.
Okay, well, we've seen plenty, so I'm assuming they'll still see them.
Sure thing. Full wide, please.
Full wide.
Yeah, especially if if it's species agnostic, we might push ahead. We're actually probably gonna have to stop the ship anyway pretty soon and let it swing for the next watch. A long bamboo coral here.
Might need to
There's a good coral.
I'm gonna swing over a bit.
It's probably 1 meter plus long.
Oh, wow.
Some really long guys here.
It's like one of those rando long arm hairs you get sometimes.
This is another bamboo coral. The long unbranched forms.
Very cool. Oh, I'm gonna start to feel a tug pretty soon. All right, Chris, you wanna go ahead and pause the ship moves?
Roger. Bridge nav, hold position.
Thank you.
What's the difference between branched and unbranched coral?
So this is one of the specific characteristics for bamboo corals that we look for. So a lot of corals can be branched, but with the bamboo corals, where they branch and how many times they branch can be one of the diagnostic characters for identifying them to genus or species. And so if we were to remove all that tissue from the outside of the skeleton, the reason they're called bamboo corals is they have these calcite segments that are separated by a proteinaceous little node. And whether they branch at the nodes or between the nodes or from a single point or multiple points are all diagnostic characters for the different types. So when we're looking at them and saying this is unbranched, there are several different species that can be completely unbranched throughout their growth.
I think you're tugging.
Yeah, I'm trying to come around to you a bit here.
Are you spinning to get the wrap out, or are you trying to get in front of me?
No, I'm trying to get in front of you. I'll probably give you a tug for a minute there.
You're more to the right of me than anything.
Yeah, I'm trying to come over to the left there, see? And then, like, lateraling, and if not, I'll try to back up. Change my heading to be more towards you. Back row, we're just getting the vehicles a bit situated here. So we're going to lose contact again for the ground.
Thanks.
Yeah, there you are.
All right. All right, let's go down together again. We just about made it to waypoint 3. That's good.
So if you're just joining us, we are wrapping up our second watch. We actually just started our expedition exploring seamounts in the Johnston Atoll. And so we recently mapped this area. It's an unnamed seamount. And we're currently exploring this area, we're exploring the biological communities in the area and the volcanic formations found here. So stay tuned and send in your questions, and we'll continue to answer them and share our experience here on the Nautilus. Thanks.
morning.
Good morning.
Everyone settling in?
It's a little early, but you know.
I think we're going to follow the last watch's plan and kind of follow along this ridge upslope on this side, on the north side of the ridge, and try and move briskly through this sediment and nodule patch. We've already sampled a number of rocks and nodules so far, so if there's anything here, I don't know what speed they were transiting at recently, but kind of following the same speed that they were using before seemed pretty good.
Roger. Sure. Sure. They were doing, yeah, that's fine.
So for those at home, we just went through a watch change. We had a new switch of our control van team and have a new group of scientists and a new group of pilots and engineers in the control van. We can start off with a round of introductions if everyone's willing and able. We'll start in the back row while the front row starts to...
You gotta talk on SPL, man. I can't hear you.
I am. Oh, you're talking to...
Yeah,
If you just push it down, it should activate.
yeah, down on the SPL switch.
You want to go first, Matt? Hi. I guess we're going to start off introductions. I'm Matthew Schaefer, and I'm one of the scientist interns.
I'm Brandon Scott. I'm secondary scientist and going to be focusing on geology. I'm a master's student in Las Vegas at UNLV.
And in the watch lead seat, my name is Steve Oskovich. I'm the biology science lead for this expedition, and I'm also a postdoc at Boston University, where I study biogeography and ecology of seamount environments, and especially deep water corals. To my right, I'm joined by...
Aloha kako, everybody. Mahalo, and my name is Kukui Gavigan. I am an ocean science intern, and I'm working as a data logger this watch, and I'm also a student at the University of Hawaii at Hilo.
Anyone in the front row willing and able to start off? Intros?
I'll go. Dave Robertson, lead video engineer and video trainer for this part of the watch. Next to me is the guy that I'm training. Go.
That would be me. Hey everyone, my name is Dan, and I am a video engineer on the 48 watch here.
Hey everyone, I'm Gibson Clark. I will be the Atalanta pilot on this watch. I'm an ROV engineering intern.
Hey everybody, I'm Lynette. I'm the navigator.
Kind of see this twiggy structure in between all these cobbles on the seafloor. I want to take a look at that if we have some time. So we're diving at an unnamed seamount, we're calling unnamed seamount 3, in the extreme southeast of Johnston Atoll region. This site had, until recently, never been mapped and never been explored before, so we're getting the first eyes on what the seafloor is like at this site. We're diving on a pretty prominent ridge, which is why we're finding a lot of rubble and debris, but we're also finding some corals, and I don't know if there's been a whole lot of sponges yet, but it's something we expect to see as we move shallower. But right now we've seen a lot of dark iron manganese rocks and most likely basalts of volcanic origin in this site. And we're going to take a look at some of the biology that we've been seeing over this dive here. Right now we're going to take a look at what I think is a bamboo coral, unbranched bamboo coral, just to calibrate ourselves. We've done a fair bit of sampling so far this dive, mostly rocks and...
Dano, you need to speak up a little bit.
Yeah, sorry I wasn't on SPL, and I wasn't forced talking to you guys. Yeah, you can zoom in if you want.
Yeah, go ahead. So zoom in, the joystick is proportional. In other words, the harder you press it, the faster it goes. Now check your focus. Hard to see on that little guy with that background. Yeah, now zoom in a little ways further.
When's the last gauge check they did?
Last gauge check was 310. Coming up on an hour.
Push it away, it looks like.
Do we have any idea what type of coral this is?
Yeah, this is almost certainly a bamboo coral. So it's kind of hard to tell when they're this deep. They tend to be very fleshy. So the one thing we can look at to kind of make that determination is look for the nodes that are present in bamboo corals. And there's one right here at the bottom of the screen.
Copy paste.
And possibly one right here. They're kind of these brown segments in the skeleton that kind of looks like a bamboo plant.
You can copy the whole
if you were to remove the tissue, but these are pretty fleshy when they get quite deep here. So this kind of coral is pretty characteristic for this environment. We see a lot of bamboo corals and golden corals this deep.
I think we want to go that way.
Can we tilt down to the base and do a quick shot of the rest of the colony, and then we can pull out and move away?
We'll have to wait. The boat's taking a runner to the southeast.
There's also a bunch of other small organisms here. These things on the sediment in the background on these nodules are probably Zenophyphores, which are small.
Okay, I can push in there a bit, video.
Small single-celled animals that make these sediment houses. So you can see another one of these node regions on the bamboos, kind of darker than the surrounding skeleton. In some cases too, if you look closely at the polyps, you can see these what look like shards of like sclerites that are sticking out from the polyps. Those are really useful for diagnosing different species.
Mover 315, 40
Are
meters.
those nodes at like predetermined lengths?
Yeah, she did this.
It's not clear. They seem to be variable as they appear. It's fairly regular, but they can be variable. You can have them very close together. I think it just depends on how it's growing at the time that starts to create those nodes. All right, all set. Front row, thank you.
It's early.
Move on.
Roger.
So does the coral feed off of those kind of single-celled organisms, or does it feed off something else?
No, most of what the corals are doing here, the entire group of corals we're going to see are called suspension feeders. So they're feeding on all the floaty material that might be around in the water column sinking down. If you look closely as we zoom out and move forward, you'll notice a gentle snow. This is called marine snow or detritus. The foraminiferan critters or Zenophyphores, they're usually feeding on dead and decaying material, either in the sediments or whatever happens to fall down as well. But the corals are actively trying to capture particles out of the water. and the Zenophyphores are more passive. There's another example of a kind of probably a baseball-sized Zenophyphore that get kind of big. They're one of the biggest sources of structure in the deepest parts of the ocean where there's a lot of sediment. It's fairly flat. So things like fishes might lay their eggs in and around Zenophyphores or some invertebrates, for example. Good structure for animals who are trying to keep their
so looking at Atalanta
generations
comp,
away
50%?
from predators.
You can see pretty pronounced ripple marks in the sediment too.
The ripples are interesting. Yeah, where you do have sediments here, that's not like a sandy beach that you might have back home. Oftentimes the sediments here are made from biogenic sources. So things like foraminiferan shells. So these are single-celled protist organisms that live up in the water column, and then when they die, their shells kind of just fall down to the seafloor and accumulate over very long periods of time, and that's what makes up most of the sediment here. It kind of looks like small spheres if you were to hold it in your hand. It looks pretty muddy right now, but it really has not a lot of really fine-grained stuff in it. Some of it could be also detritus and dead and decaying material.
Yep. $12 meters a minute.
Just doing a few tests here, Steve, while we're waiting for the boat. This is going to be Herc crashing into the seabed.
Got it. Looks great.
Seabed crash test complete. That's a bug in the code that still gets me to this day. The first time you engage auto altitude, it drives the vehicle hard into the seabed. Yeah, I didn't catch that till too late. Sorry. I'll try that again. Oh, it's going to do it twice in a row. Look at that. Yeah, once wasn't good enough. I would have thought by now, 2 watches, we would have had that bug. So it's a bug in the software. You have to double tap auto altitude. So along with that, it looks like Gibson we're approximately probably 35% thrust to keep Herc. Let's just try it with the Z-bias. Sure. Okay, Gibson, you can come up 5 meters on the winch there.
Coming up 5 meters. How's that?
That's good, yeah, thanks. Are you zoomed out on Argus video?
Yep.
Yep, all out.
Roger. Looks like they've completely shredded the... Oh, they've dropped 2 of the weights. That's what. That's interesting.
Okay, gauge checks are complete. I can reset this tension here.
Yeah, roger. You can, this thing.
If I want to toggle that tension back up.
You can, yeah, it's there somewhere.
Okay.
It's right here on the...
Grafana.
Yeah. Doesn't need to be there in the...
Okay.
We're well within the box.
Any indication of other relief coming up, possibly, or is it just this kind of stuff for a while?
They're not...
Yeah, I think it's maybe this kind of stuff for a while.
Okay. Kind of move briskly through it then.
Yeah, we can kick him up to... kick her up to... Let's try half a knot for a while.
It's like some fallen debris, sponge debris maybe. Stalked crinoid, type of echinoderm, sea star relative.
That's why. Oops. I'm gonna display with the lights here for a minute. That's the screen you want up there, that Grafana screen.
Right here on the left?
No, you want to keep an eye on this. Yeah, what you're watching there is that delta. If you go to the one that says pilot less.
I like that one more.
Well, there's a reason for that when you get there.
There we are.
The delta will change color there if it gets out of the box. And then this also gives you an idea of
yeah, okay, the relative depths.
yeah, you get more of a kind of a visual there. And then this one will be the winch, the vertical velocity history, which I don't need to worry about because that's all over at the moment.
So we're kind of been moving in and out of these kind of rocky, what look like rubble, cobble-sized, terrain, volcanic rock terrain. I think we've passed through a patch of sediment, probably a bit of a lower slope on the ridge here, but it's starting to pick up again, it looks like.
I can come up 5 on the winch there.
Coming up 5.
So your first cue is going to be keeping her, should be in the middle of the screen. That could be me. It's most often me flying out.
Okay.
But when you see it getting low like that,
yep.
And if the delta is in single digits,
yep. Want to come up?
Yeah. You can come up or let me know, and I'll ask you to come up.
Roger that.
So the main thing you're watching to begin with is that delta.
Gotcha, yep.
And then the other one is your Argus altitude, which is this guy here. And then the third one is going to be the sonar. If there's any returns within that first 20-meter range ring.
Yep. Give us a holler.
Yeah, let us know about it.
So what you're seeing on yours is the seafloor there.
Yeah. Yeah, you can do 100-meter moves if you want. We're just now starting to move, so.
So we were on the first dive of our about 3-week-long exploration of Johnston Atoll. We're going to be focusing on areas around the southeast of the Johnston Atoll region, a little bit in the west of the Johnston Atoll region, and maybe Johnston Atoll itself as weather and time permits, doing shallower dives there. But for now, this is kind of the environment we expect to see for the next few weeks. We are deeper than where we find a lot of high-density coral and sponge communities, which is probably why we're not seeing as much, and they're pretty sparse. But being deeper also helps us get good quality basalt rocks for dating some of these seamounts, which tells us something about their origin. Brendan, are you going to be working on these particular collections eventually?
No, I'm not actually. This is all Kevin's
come up 5 again if
work.
you want.
Yeah, he wants to date these
coming up
argon dating. I'm just here to sort of understand the process of sample collection and the environments that we see these basalts in.
What was it saying? Like good rock, bad rock? Like which one was better?
Right, so for sample collecting, a good rock would be something that's angular, because more organic material can get the ferromanganese on it, and those appear more rounded. So just to avoid that, because we can't see the internal contents of the samples until we cut them open, we want more angular stuff, because that's how the rocks form naturally.
So at 20 meters out on the sonar, I'm picking up Herc, hey?
No, you can't really see Herc at the moment because I'm...
So what's that feedback there, like 25, 30?
I'm underneath. That's the slope.
Oh.
One of the things we noted with our dive planning for this site is that we suspect that the eastern side of the seamount was subject to possibly faster current flows than this side, which would explain why there's probably a lower density of animal life here, suspension and filter feeding animal life on this side. But due to kind of permitting and planning restrictions, we're kind of limited to working here because we're right on the boundary of the USEEZ and international waters. So to dive on the other side of the seamount would kind of be outside of the objectives of this cruise. So it'll be a good comparison. We have a few sites that we're going to be visiting tomorrow, not too far from here, about 30 miles. That would allow us to do a look at a different aspect of the seamount on a geo type seamount, which is a flat-topped seamount, probably exposed at one time at or above or near sea level in its geological history. This one doesn't seem to have any evidence of that based on the mapping we've done. But we're seeing a lot of dark iron manganese crust. Makes these rocks look really smooth and loamy, which can sometimes be a challenge for collecting rocks as well, because a significant portion, sometimes several centimeters of the rock, can be crust, and to get a good rock collection, we need to get at the stuff that's inside.
Some scientists could probably use a big amount of crust if they wanted to analyze it, but for our goals, that's not what we want. I'm probably going to cut all that off when we prepare the rocks. And Steve, you were talking how later on we'll see a geo and potentially near the surface. A good way to know if it was near the surface is if you see any carbonate rock around there, because the corals will, sort of live there and die there and lithify into limestone. And if we see evidence of that, then it was most likely near the surface, because you need a high density amount of coral to create the limestone.
Come up with another 5.
Coming up.
Picking up a little bit here. How many samples do we have so far? I know we have one water sample.
I could tell you how many rock samples we have.
We're on sample number 7. Like we've So I assume we've taken 7?
Yep.
I believe that is correct.
Mostly stuff in the starboard bio box, rocks, and some nodules from earlier. One Niskin water sample from a kind of a background water sample for our environmental DNA analysis of this site. I'm going to keep an eye out for higher density communities of corals and sponges and other benthic biology to do more sampling of the water column here.
And I believe we also have 1 fossilized coral on board as well.
Oh, cool. Another large octocoral colony coming to view. This one is almost certainly a bamboo coral as well, probably similar or the same type species that we've seen in the past, just a bit larger. It could be different though. It's hard to tell in some cases because oftentimes you have to do a quick snap zoom on the polyps to see if they bear any similarity to what we just saw.
Snap zoom there if you want.
Yeah. But you should be able to keep an eye out for those nodes, kind of these dark bands. This one actually is different than the one we just saw. Still pretty fleshy, high density polyps up and down the axis. These are usually types of corals that we'll see getting sometimes 1 or 2 meters tall. Oh, there's some sort of gelatinous plankton in the background too, maybe a ctenophore.
Do we know why it's like that kind of color, that kind of fleshy color?
Not exactly clear. So bamboo corals can be a lot of different colors. Usually they're translucent, but sometimes they can be pink or opaque. You know, the deeper you get, typically the more, you know, tissue they have. So they, you know, it kind of blocks the skeleton. It's hard to see the skeleton, but the color can be variable. You know, it could be something in their diet or something that they express. You know, from their ancestral lineages.
So then something like black coral or gold kind of coral, like, is that just like what they eat or is that just their environment?
Yeah, for black corals, it's a little bit more restrictive. So black corals will often have a black skeleton that's made out of a protein that they produce for their whole skeleton. That they really don't produce any calcium carbonate. In contrast to, you know, a lot of the corals we just saw, like the octocorals or even some stony corals. So they have a little bit different growth patterns.
And then that thing that's just coming up to our left, what is that, would you say?
We'll take a look here. Do a quick zoom. I don't know if we've seen any of these yet.
Enhance.
It's a BBC shot. It's all. Hey, Dave, can you put the DSC up in one of those black holes there? Or Dan?
What is it, Dan?
I want the digital stills camera in one of the.
I don't see it. We don't have that as a source.
They were saying there was a ground fault on it earlier, so they turned it off.
Really?
Yeah.
Okay.
You can try it again if you want to, I guess.
You can zoom in there.
But I haven't been using it back here.
Right.
All right, so we're going to take a look at this
Push in a little more if you want.
octocoral. From a distance, it's hard sometimes to tell the bamboos and the primnoids apart. But as we get closer, we can start to make out some characteristics. This one looks to be a primnoid. So you can see the skeleton is fairly uniform in color. Looking through at the axis, the skeleton material that forms the branches, you don't see any very obvious nodes, but there are branch points. Given the branching sequence here, and then the shape of the polyps, we can say that this is in the genus Norella. Polyps appear to be pointing downwards, and the branching is equal dichotomous from the bottom. So that just means that from one branch point you get, 2 branches. So that's pretty characteristic of this genus. To know more about species, we would have to take a collection of it or look more closely at the polyps. Sometimes you can actually get really, tight zooms and actually get a look at, sclerites that are present in the body wall of the polyp from the ROV, but it's pretty rare. You have to be really well lit and tight up close on them.
And then that little lollipop looking thing, do you know what that is?
Slower back down to 0.3.
Sponge.
I'm not an exact scientist, so lollipop looking thing will be my descriptor.
To quick zoom on the sponge there.
Also okay. Yeah, this is a glass sponge. Oh. We would need to rotate around the back or, to the side to get a look at the other side of it, which tells us something about its morphology that could help us get a little bit more refined identification. But we know it's a glass sponge. That's typically what we find down here. So this looks like it could be maybe Colophacus. See how it kind of attaches to the back there.
Zoom out just
Looks
a little
almost
bit.
fungal-like, which is kind of cool.
Mm-hmm.
Cup coral in
yeah,
the
these, unlike the corals, these are filter feeding, so they're generating their own current using cells inside of their mass that creates a current and allows them to extract nutrients. That's great.
Is that a cup coral there?
Oh yeah, what do you got there? Huh.
Oh, cup coral.
You know what?
Quick zoom on that guy.
You know what this is? I'm glad we have the scientists ashore, because this is something we collected back in March in our Kingman and Palmyra cruise, in Papahanaumokuakea last year in November. We collected this and we were trying to figure out what it was.
Okay,
we thought it was an anemone of some type. All the cruises are starting to blend together for me now.
Me too.
But we collected that, and it was an anemone of some type. We suspect, but it's good to know, an observation of that animal gives us some idea of how widely that species might be distributed in the Pacific.
It looks like something at a dune.
Yeah, it's strange. It's anemone-like, but it didn't contract like an anemone.
But that was definitely
So is it like an
Nigerian,
entire
right?
subspecies entirely?
Yeah, most likely, yeah.
Or coral.
Let's stop her up for just a minute, Lynette. Sorry. Yeah, I'm out of the box here. I need to. Not that Atalanta will hold position, but at least it'll give me a few more seconds to. Sure enough, Steve will see something he wants to see. Well, I'm on the wrong
always
side.
Things to see. It's a balance.
Yeah, I spent a little too much time there and got on the wrong side of things. Okay, Gibson, you can come up 5 there again.
Coming up.
It always happens right when it starts to get interesting. That 0.5 gives us a bit more of a layback as well, so if we were going to stop and sample, we basically have the sample time of Atalanta running over us. you pay for the speed.
Just watching our delta at 7 and come up a little more.
Yeah, you can come up a bit more as I'm coming underneath you there.
Roger.
You definitely don't want to let your altitude come into single digits.
Roger that.
Weather's somewhat benign, so we're flying pretty close here. But usually when I come under, we want to have a little more
Steve is predisposed, but looks like another bamboo coral would be my guess, my extremely novice guess.
I want to say you are correct.
I'm just going to be a sheeple and say that you're also correct on that one.
Okay, we have 100% confirmation from people who are not biologists.
Not the experts.
I can tell you we're looking at some low bait flow, basalt flow, below the coral, if you're interested in that.
Please enlighten us on all the rocks around us.
No, that was pretty much the end of what I was going to say.
Oh, okay. Well,
there's not much detail to go into in terms of flow type.
Mary, I like that. I think a bamboo whip is probably the best type of description for this.
Would you mind sharing with us how these low bait basalts form?
Yeah, so at some time or another, we're going to want to get the exact age of when this occurred, but magma effused out from some conduit,
now.
And depending on how fast it was flowing out, you get different flow types. And it'll solidify rather quickly. Oh, there's a sea cucumber.
Oh, Liz can like that.
A grape flavored one.
Quick zoom on the sea cucumber there. Can slurp that guy up if we want.
Yeah, what did we,
10 seconds.
How big is that?
Decision. Yes or no? 9, 8.
I don't see the lasers, sorry. Oh, yeah, they're there. We'll pass on this one. We've been seeing them pretty often.
Okay, go on. Atalanta never stops.
You win this time, sea cucumber.
What's your transit speed?
We backed off to 0.3. It was a ridiculous layback there.
Sounds good.
And it's starting to get a little more interesting here. It's coming a little more steeper, or at least it was.
There's a question asking about the origin of the lava flow. We're not sure, but around Johnston and the Line Islands itself, the origins are deep mantle for the most part. But you never know until you do chemistry and we get the ages on them.
Coming up.
My guess would be deep mantle from a mantle plume, but unknown.
I was kind of expecting these seamounts to be, given their age, to be more, densely coated with crust, but it seems like there's a fair bit of loose material, which is sometimes a challenge on these seamounts, especially the older ones. Everything is cemented in with crust. Oh, yeah.
Sure.
So there's a question from the shore for the front row about how do Atalanta and Argus compare when piloting Hercules? Is there any noticeable difference?
Yeah, there sure is. Atalanta is all over the place. It's not as predictable as Argus. It's less of a weight on the pendulum there. Seems to be more affected by currents. It bounces around quite a bit more when the
Sure
ship
does
heaves.
bounce around from the camera angle view, I'll tell you.
Yeah, it does. But we're not at all worried about the weight on the wire at the moment, so.
Great.
It's a trade-off, I guess.
So you can go deeper, you're telling me?
Yeah. Deeper till it hurts. Do a snap zoom on.
Yeah. So this critter, I don't know. I have wagered they've probably seen some of these so far. Oh, there's a tiny little
how many baskets
isopod there or something. So we've got a small isopod here, crustacean. We've also got this big red critter here. This is a sea star, actually. Sometimes they're often confused for crinoids, but this is a Brisingid sea star. So a sea star like you might see at the beach or intertidal zone, but this one is really unique to the deep sea. They have these large spines coming off the arms, and they kind of adhere to the rock surface, and they use their spines to pick out particles and filter out particles. It looks like one of the arms is damaged here, but they will often be observed to regrow those. So pretty characteristic, common species for this depth. Sometimes you can find them attached to rocks, and sometimes they're attached to corals or sponges and things. So
don't the sea stars usually start eating sponges and coral? Am I wrong in that?
Zoom in there, video.
Some of them. So this one goes about life by picking out particulate matter from the water. Oh yeah.
Give it all you got there.
Isopod there.
And name him.
That's on X.
Got full zoom in?
So this is an isopod, or on land you might call them roly-polies. A lot of species in the deep ocean, including the giant isopod, which is not usually seen around here. It may be found around here, but I haven't ever seen one. But this one is fairly large for its size.
For a second, video.
How big can isopods get?
About this big, like a football sized, a little bit bigger.
If I can get a little better shot here before I get drug away.
Yeah, the giant isopod was commonly observed when we were doing explorations in the Gulf of Mexico, for example. They're sediment dwellers and detritivores. They eat dead and decaying material on the seafloor. They can go very long periods of time without eating. Very large compound eyes. It really looks alien.
Okay, I gotta catch up again. Sorry, Gibson, usually I can stay in the Argus view, but yeah. You can come up another 5 if you want.
Okay, coming up.
faster. Single digit altitude there. Yeah, that should be good. See that curly Q in the tether there?
Yep.
Yeah, I'm living right on the edge. Okay, come up another 5 meters.
Okay, coming up.
Back in the box.
All right, our depth right now is 2720 meters. The top of the seamount peaks at around 1800 or so. Although I suspect, we'll probably see more biology in the next few hundred meters as we start to move into the deeper diversity maxima for this area, around 2,000 to 2,400 meters. We've been having some really good signs that there's more diverse biology coming up. Seeing more species that we haven't observed down deeper. What do bottom currents feel like here? Sluggish or?
Yeah, I haven't really, haven't noticed any sort of current, any significant current.
Another bamboo coral, unbranched.
Oh, there's something over there. A shrimp, maybe? What's what it look like?
Yeah.
Yeah. It just kind of bounced off the ROV.
Yep.
Hey Dan, let's try a zoom, but this time do a nice slow zoom. There you go.
Yeah, so
That's
this,
good
the shrimp in the background looks like nematocarcinus or nematocarcinid. It's a long-legged shrimp. Usually they're found in soft sediment type environments, but they have these really super long, fine legs for walking on soft sediment.
He looks like a Craig.
Yeah.
And what genus was that again, you said?
I think it's nematocarcinus. N-E-M-A-T-O. Carcinus. Yeah.
Okay, you can go wide. So yeah, that's a nicer speed. When you zoom really fast, it's a bit disorienting for me.
Copy that.
It's like putting your face to the window really fast. When you're zooming in slower, a lot of times I'm still moving the vehicle while you're zooming. So as you're zooming in, I'm trying to position the camera to get the shot.
Some folks on shore were asking about the clarity of the water around here. We are pretty far offshore, so oftentimes the distance we can see with our lights is tens of meters sometimes. Oh, a very large bamboo colony there. But we're also in what's called part of the oligotrophic ocean, so that means nutrient poor part of the ocean, and typically in these parts of the ocean you don't have as much productivity as the continental margins or the equator, and so what we have is relatively clear water because there's not as much productivity, but there's still plenty of food for lots of deep water animals like these corals.
Got a wall
and
on the
sponges.
sonar pretty close here, Dan.
Yeah, good eye. You probably want to come up.
Coming up.
I'll go check it out.
Delta's at 20 meters.
Yeah, I'm, the delta's kind of, I don't know how I want to explain that.
Anemone down there.
That altitude, we
Yeah.
definitely want to.
Roger.
Delta changes as since we're going up the hill, as I stop and Argus keeps going.
Yep. I can come up to that hill a lot closer.
Yeah, we definitely want to
keep that double digits.
Yeah, keep it double digits and keep
So
the color out of the 20-meter range.
There's a question about asking about whether seamounts can form just from magma sources, or if they can maybe form tectonically. It's most likely a mixture of both. I mean, obviously, the volcanics have to form volcanically through magma effusion, but the height in which they reach can sometimes change if you're near a tectonically active area, like plate boundaries, which we aren't near right now. We're in the middle of the Pacific Plate. And most seamounts are volcanic origin, but you can find non-volcanic near those tectonically active areas.
Looks like we have another sea cucumber down to our left, but it's now gone. But
sorry, I missed it.
oh, no
Oh
problem.
yeah, right there
it
in the sediment. I'll circle it here.
Oh, I see it now, thanks. Wow, speaking of sediment. Too far side onto the hill.
Yeah, the next sea cucumber we see, we should slurp it up if we're in a good position. So keep an eye out so we have plenty of time to stop. But I don't think we're going to be seeing very many on this wall. So
I never thought I'd heard the word sea cucumber and slurping it up in the same sentence.
That's the best way to get them.
Suction sample, a more appropriate term.
It looks like we might be in a sheet flow now, which is pretty interesting. The flow rate was probably higher and it's more extended rather than disconnected like you see with pillows or low bait flow.
And this is more glass sponge that we're seeing right here?
Yeah, it looks like another glass sponge, probably also Colophacus, similar genus to the one we just saw, a bit deeper. My sponges aren't as good as my corals, so I'll be relying on folks ashore and our handy guides here to help us out.
That's true, we have a little cheat sheet.
Now we're back in a pillow flow, so I mean you can see it's pretty interchangeable, the flow type.
So then how do these things kind of come to be? Is it, there was plates here, there was some sort of current that pushed all the magma up? How does this form?
So I mean, it's magma. You're right.
Come up
And
5.
it pushes up because of convection.
Yeah.
The heat exchange. It wants to
Can
move up.
do a slow zoom in
And
on
some
that
kind of conduit was
sponge there?
opened.
Yeah. This is a different type of glass sponge.
Good there, thanks.
Oh. Craig's back.
This one looks familiar, but
slow her down to 0.2, Lynette. I can't seem to stay in front of it.
Is that another
Bridge
shrimp
nav?
living inside of it?
yeah, a lot of the times these glass sponges will have
Can we slow to
associates
0.2
living
knots,
inside
please?
and around.
Thank you.
So you can see a couple of shrimps there. Sometimes they'll have amphipods or isopods, crabs, squat lobsters. Great. So all of this mass of this, oh yeah, there's another shrimp there on the right-hand side. There's all of these fibers are actually glass
coming up on 22.
fibers, glass spicules that are
okay, can go wide
weaved
now.
together by the sponge. Thanks for the imagery there. I think this one might be a rosellid as well. Family Rosellidae.
How you doing over there, Gibson? Getting comfortable?
Yeah, doing good. Gonna start gauge check shortly. It's just still 500.
Yeah, Reg. Let's try tilting that camera down a little bit.
Roger that. Getting funky readback on the Argus altimeter. Jumps from 10 to 40.
It's jumping around. As it often does.
You want to pan the Atalanta camera?
It only tilts, so you can tilt down. Tilt it down to -90.
-90.
Since I seem to be spending all my time under Argus tonight.
Just gonna winch up a little bit here.
I don't think there have been very many fish seen yet. So we saw some Zenophyphores earlier, the eels.
Saw a couple of Halosaurus early in the day.
Yeah.
Zoom in just a bit for me, Dan.
Halosaurs, typical for this depth.
That's good.
So that rock's got a bald patch.
Yeah, I'm not sure what's going on there.
That's good on the tilt there.
Roger. We're at -90.
Roger that.
Starting gauge checks now.
Just a weird looking pillow.
Yeah, just run through them as quick as you can. So start hitting buttons.
A lot of both the corals and sponges we're seeing at this depth are probably fairly old, you know, at least decades, I would wager. It's really hard to date a lot of corals and sponges that are this
next
deep.
button.
It's going to come up a bit.
You know, using our scientific means, so we often have to just rely on visuals and sampling, which we're trying not to destroy whole colonies if we don't need to. You know, to find the ages of things. So we're kind of reliant on just using colony morphology and size to approximate how old they might be.
Check.
A lot of corals and sponges will grow fairly slowly, so we can estimate that the bigger ones are probably quite old. Got a couple of things here. Another unbranched bamboo coral, and just below it there was a yellow stalked crinoid.
Really?
Yeah. Really fine critter.
Oh yeah, good eye.
and another
Let's,
crinoid to the
zoom in
left
there.
side as well. Yeah. Just fell off the rock
That's
there.
good, thanks.
These are the two different types of crinoids we typically will see. The stalked crinoids, which are fixed to the rock surface, and then the comatulids, which often are detached and free living, free swimming, can move around, can swim up into the water column, glide back down.
That's good on the gauges then. You can put the camera back to 1.
That's HV comp,
What's that?
high voltage,
Yeah.
and HPU.
So how long does it
Thank
take
Motor,
the average
HPU.
coral to grow, like per year?
I
or
know
so?
the res is good, so.
A lot of that will depend on the which species the coral belongs to, which family, what depth it's living at.
Good on the
obviously, the shallower you are, the faster you're going to grow because your food quality is a little bit better than down deeper. The deeper you are, the poorer quality food you have. That's because all the food sources from this environment come from the surface ocean, and as that dead and decaying material, marine snow, sinks through the water column, it's being consumed by bacteria that consume the carbon out of that food product. And what's left is, when it gets down after sometimes taking years to drift down to the seafloor, it's very, low quality, low carbon food source. Oh, there's something else right there.
So
that
is
one
that
might
why
be new
you
for the
see
dive.
coral more towards the surface, more vibrant and bigger usually?
Bigger, faster growing, yeah, sure.
Okay, Dan, you can zoom in on that guy.
So we got a black coral here. I think this one might be the
Zoom
first
right
one for
in
the
tight on
dive,
that guy.
but this one looks
Oh
like
yeah, look at that.
heteropathes. Yeah, you can see the black skeleton right through the tissue there. Tissue is kind of dark or reddish. Oftentimes through capstone we were calling this heteropathes americana or something similar. But you can see the morphology of these polyps is slightly different than the octocorals. The mouths of the black corals are actually
Don't move the winch while we're zoomed in.
after every 3 pairs or 6 tentacles. So they're hexacorals, so they have that six-way symmetry. So oftentimes what you'll see is there'll be a mouth here, 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 and there'll be 3 pairs of tentacles in between each one of those. So the kind of darker red bits, those are the mouth parts.
So the one that's to the left, of course we can't see it right now, it's missing a bunch of stuff on the top. Is that just because it was underdeveloped or it was damaged, or?
That one, yeah. So that could have been predated upon by some other species. Could have been grazed. Oftentimes you'll find some small invertebrates will graze parts of the coral, and then either the coral will regrow, or sometimes you'll have opportunists come along, like hydroids, that might land on the bare skeleton and grow.
Yeah, you can come
It
up
feels
now.
like the Charlie Brown
Fun.
Christmas
Yeah,
tree of
coming
coral. It's a little sad.
Coming up, watching
That
the
one looks
sonar, I
pretty
got
good,
something close.
but you'll see some like that, yeah.
Looks like a big wall in front of us there.
Yeah, it's a bit of a rocky point there.
All right.
It should be good there for a few minutes.
Raj. Did our comps on the Atalanta camera shift?
No, and we just, since you tilted the camera down, it's in the way of one of the comps.
I see.
But you see how far they dropped from where we started on deck?
Yeah, it looks like we're up to 50%, maybe even, yeah down to 50%.
Somewhere in there.
It's consistent.
That looks like another stalked crinoid below there. I think we may have passed it.
Yep, it looks like it.
for another 5.
Coming up.
It's getting pretty steep here if you're looking at the Atalanta view, which is at channel 2, I think. You can see how quickly it's rising. The seafloor.
You should be able to tilt up a little as I come back out in front of you. It's where it should normally be, but I can't seem to stay out there tonight.
Oftentimes, this kind of looks, somewhat, loose and unconsolidated. You won't find very many corals and sponges in this type of habitat, possibly because these rocks might not be very well held into the side of the seamount. They might be subject to falling. For example, if there's some sort of disturbance. So you often won't find large corals and sponges growing on these because they're subjected to more frequent disturbance. How do you feel about a collection of this cucumber?
Cucumber.
Sure.
Ooh, great flavor.
Yeah. So if you change your page to the Herc hydraulics there.
Raj.
And change that camera to the jar as we did earlier.
What's that?
Perfect. There's nothing in jar 4, right?
Nothing in any jar yet, right?
I believe so. We have room for 7 slurps.
I love that we're calling it
Dan,
slurp.
did you see we had a glitch in the Herc camera?
I did not.
Okay, I think it was when he switched cameras in the...
Yeah, it was, yeah.
Yep, I thought we had solved that. Darn.
Okay, you see where it's I'll do it the first time. The spare,
Okay.
I think this one is actually suction. I never did relabel that correctly.
Oh, okay.
This one's not suction.
Roger
so
that.
See how I've cranked it up to 100% there?
I do, yep.
Yeah.
Gotcha.
Video, you want to zoom in a bit there?
You want me to shut it down once it's there?
Yeah, once it goes in the jar, you can.
All right.
Now you can hit 0.
Roger that.
Great. It's a good collection, sea cucumber, for understanding deep sea food webs.
I feel oddly guilty.
This one is some type of synallactid. That's the family. Maybe Paleopatides here.
And this is in jar 1, right?
Jar 1, that's right.
Got something steep right in front of me here. I'm just going to pull up a little bit.
Roger.
I think it's this one. For the identification, we can write this family here.
All right, I'm clear.
Yeah, there was a interesting wall we were just coming up on there.
So this tension on the checks here, this is max tension every hour, is that right?
Yeah, you can pencil whip that one.
Is that another sea star right there that we see?
Yeah, it's another Brisingid. Yep. Yeah, we'll start to see kind of the more typical sea stars for this this deep water environment. As we get a bit shallower, we'll start to see goniasterids, which is sometimes known as cookie stars too, will start to appear.
You can change that camera back to the rail cam now.
Okay, copy that.
And what kind of glass sponge was that we passed over?
I think that was Bolosoma or something in the Bolosoman family. Definitely a euplectellid, so it's in that family. And then possibly Bolosoma species.
To, go ahead and come up 5 there.
Coming up 5.
That was what we call a Dr. Seuss sponge.
Why do we
Technical
call
name
it
for
that?
No idea. One of our more famous geologists we've had out here named them that, and it stuck.
1 sponge, 2 sponge, red sponge, blue sponge.
Something like that.